that has been put on by the Canadian Pollination Initiative on bee-vectored biocontrol technology. This is a new technology. Well, it's not that new now, but it's a new technology which is becoming commercialized slowly in various places. And really what it does involve is pollination and crop protection taken together. We're very pleased to be able to acknowledge the Natural Sciences and Engineering Research Council of Canada for its support to the Canadian Pollination Initiative, and also for this particular webinar, the International Union for Biological Sciences and the International Commission for Plant Pollinator Relations. And they have been working closely with the not-for-profit research and development organization, the Arthur Dobbs Institute, which has helped put this together. So with that, I will introduce Dr. Les Shipp, who's going to talk to us about how this bee-vectored biological control technology has been used successfully in the greenhouse industry. And following that, I will follow up with a brief summary of the sorts of things we've been able to do with open-grown or field crops, particularly in Canada, and some of the new initiatives we're taking on. So thank you very much, and leave it now to you, Les.
Thank you, Peter. And I also like to welcome everybody to this today for the talk I'm going to give on looking at greenhouse pollinators as a new delivery system for microbial control agents for greenhouse crops. As you can see, it's really been a team approach with people from the University of Guelph, graduate students Jean-Pierre Kapongo, Mohamed Al-Mazarari, visiting scientists from Korea, and also we've had work with Ontario Ministry of Agriculture, Food and Rural Affairs, and with bee vectoring technology. And I also like to acknowledge BioBest for supplying the hives for the work that I've done. There we go. Why the pollinator vector approach? We all know that honeybees are excellent carriers, and they are able to pick up very small pollen grains, but also they are able to pick up fungal spores and bacterial cells. So it was actually hypothesized by Dr. John Sutton and Dr. Peter Kevan back in the early 90s, why can't we even use honeybees to carry beneficial microbial agents? So what's the advantage of using bumblebees as a novel application strategy for microbial agents? Well, the bees are already out there being used for pollination in greenhouse tomatoes and also in greenhouse peppers. So it's just another value added to the bees to combine pollination with the distribution of a biological control agent. And since the bees are out in the crop 7 days a week, you now have continuous dissemination of the microbial agent to the target plants. And since the bees are going right to the flower for pollination, you now get this microbial agent being delivered right to the flowers. But also, since bumblebees are buzz pollinators, they're grabbing that flower with their mandibles and with their thorax. They have vigorous vibrations. They are shaking off this inoculum powder onto the flowers, but it also goes into the air and it falls on the leaves, both the top and the bottom. And this is really important that it does get onto the bottom side of the leaves. So now we have less of this agent, microbial agent, now going into the environment, the soil, air, and water, because it's going right on the plant. And of course, there is a labor savings cost here because we're not spraying. We're using the bees to deliver that, so they're both environmentally and economically friendly. And of course, as a microbial agent, usually there is no post-harvest interval. We've also found they are potentially compatible with other bio-based IPM programs. Now, this slide here is just a summary of the bee vectoring technology development that we've done. You can see here we started way back in the early 2000s, and really what we did then was to take a look at proof of concept of this idea. In other words, can the bumblebees be used to deliver a microbial agent to control a pest? In this case, we looked at ligas, this is tarnished plant bug, and thrips. Then we went and took a look at the optimal concentration that you needed that the bees could carry to kill the pest but not harm the bees. And I'll show you some slides of how we combine two agents into one inoculum for both pest control and disease suppression. And we've done some commercial trials. We've also looked at the other beneficials that are used in greenhouse production to see how they're compatible. And then we looked at other agents. I'll talk about baculoviruses and Bacillus thuringiensis, and we conducted trials with them too. Now, the first agent that we looked at was Beauveria bassiana. We looked at this agent because first, it was registered in Canada, and it has a very broad host range. You could see here that it attacks white flies here. You got western flower thrips that are very well infested here. You got the tarnished plant bug, and of course this one here was the aphids. So it has a very broad host range, but also attacks bees, so you do have to be careful at what concentration you use. Now I talked to you about the other beneficials. That is the parasitoids, the whitefly. This one here shows you the whitefly parasitoids in Carcia and Eretmoceras. You will see here we did cage trials to see the impact on these other beneficials. And we had a control here. There was actually no bee just the bees were operating in here, but there was no microbial agent. The microbial agent here was Beauveria bassiana. And you could see here with the mortality between the two treatments, there was no difference. And that was both for Encarsia and Eretmoceras. Then when you take a look, the most important thing is to take a look at the parasitism levels of whitefly nymphs here. And you could see here with both treatments, there was no impact on the parasitism levels. This was the same result that we also found for Aphidius. And of course, again, we also looked at predatory mites. There was no problem either with them. They had the same efficiency and no impact on their mortality. Now this slide I wanted to show you here has aureus. And I put this up here because aureus is used in greenhouses, predatory bugs, but also Microlophus and Dicyphus. You notice here our control mortality in the trial. It was essentially about 17%. But you notice here when we had the bee vector Beauveria, it was up to almost 42%. So the aureus is more closely related to tarnished plant bug, and you can see here there is an impact of the Beauveria on this beneficial agent. But the ones that did survive, there was no difference in their efficiency. So essentially in the control, you had 19, almost 20 first instars that were consumed over a 24-hour period. And the same when you did the Beauveria treatment, you also got about 21. So it's really important, though, if you use bee vector Beauveria with aureus or any of these other predatory bugs, that there may be an impact. So you need to check this out. Now, after we did all our trials at the research center in Hurl, we did cage trials there. We went out and you had to now take a look at the commercial setting. And it was really important here to determine if the bee vectoring and the carrying of this inoculum had any impact on the pollination efficiency and the fruit yield and quality. And we found by our trials, we did this out in greenhouse tomato trials. We had about 3 or 4 growers in the Leamington region, and we found that there was no impact on the pollination efficiency, nor there was not any impact either on fruit yield and fruit quality. This trial here, this is Shalin Kosala from the Ontario Ministry of Food and Rural Affairs, out showing one of the summer students how our setup worked here. And what we found is that the bees, when they left the dispenser, they were picking up the same amount of inoculum as they were in our cage trials. And then they were transporting this inoculum down these rows here, which were 100 meters long. So you could see here on the slide, on the graphs on the left here, you notice here that when we took our leaf samples, we collected leaf and flower samples at the top half of the plants. You can see we came back, we washed these samples. You can see we washed them and we plated the diluent onto selective media, and you can see the colony forming units on here. Each one represents a single spore, and you can see that we were able to get recovery of the spores all the way down the row, right from we took samples at the front, middle, and back, and the same for the flowers. The levels that we recovered in our commercial trials were roughly about 10 times lower than we did in our cage trials, but in our cage trials we only had 64 plants in a cage, and here you can see that you've got over 10 to 12,000 tomato plants per acre. So there's a substantial increase in density, and we're still able to get transmission of this inoculum. As a result of this work in the commercial houses and what we did at the research center, we're able to get the first label expansion for bee vectoring for a microbial agent, and we had Botanic Guard 22WP, has to be a dry formulation, of course. We're able to get registered for bee vectoring in commercial crops. Now, I talked to you earlier about looking at combining two microbial agents into a single inoculum for pest control and disease suppression. Here, this shows you the results of combining our Bavaria bassiana with Clonostachys rosei plant health inoculum. And when we did that, this was in tomatoes, we were able to get 58% reduction in gray mold when we did this in the treatment here where we had bee vectored inoculum compared to our controls here, our two types of controls, heat inactivated control and no inoculum. And when we did the leaves, we're able to get about 48% suppression of gray mold. So that was good control of the gray mold. And when you went over and you took a look at our white flies, we were able to have still about 58% infection of our white flies, mortality, and there was no impact really on the bumblebees. You could see here the bees here in our bee vector area here treatment, and you can see the mortality was not significantly different to our other two control treatments. So we're able to get disease suppression and pest control at the same time. Now, I mentioned that we looked at other microbial agents. We looked at a baculovirus, and we looked at Bacillus thuringiensis too, Kerstaki. And this slide here shows you one of the visiting scientists, Dr. Chudong Lo from the Chinese Apiculture Institute, and he was helping out in trials. And these were cage trials that we did. We inoculated the plants with second or third instar cabbage looper larvae. We then put our hive in, our inoculum. We left them for 4 or 7 days. We took our samples, and you can see here, this is a cabbage looper larvae that's been infested with the virus, and then of course this is a classic symptom here with the BT. This slide shows you what we got for results. For a control, we had Dipel 2X dry flowable, which was a spray formulation. You could see after 4 and 7 days when we collected our samples here, we had 100% mortality. Then we looked at Dipel. This is a dry powder formulation, and you could see here at the different concentrations where we didn't dilute it, we diluted it by a half, a quarter, or an eighth. You can see here when we had 25 parts of Dipel with 75 parts of the carrier, after 7 days we were still getting 100% mortality. So that was very good. So after that we went out and we did some commercial trials. And this slide here shows you in, this was in a commercial pepper house here, that when we had 2 different types of dispensers. The first one here is got at the bottom, that's bee vectoring technology dispenser, and I'll show you that later. And also flying doctors, dispenser, that's from BioBest, and I'll show you them. We use both of them out in the commercial situation, and both of them reduce the populations of cabbage looper by about 4.5 to 7.5 times lower than the control treatment. And this is the number of cabbage loopers that we caught on the pheromone traps. So it did work. We did some tomato trials too, and there was essentially not a lot of difference between the control and the bee vector treatments, but they both worked the same. Now, we also took a look at a virus, baculovirus here, and this was isolated by a colleague, Dr. Martin Robinson from our Saskatoon Research Center, and we went and we compared spray treatments here. These are the full rate, half rate with bee vectored baculovirus. And again, you will notice that with the bee vectored baculovirus at the full rate, we were up to 97% control after 7 days. When we did the half rate, it wasn't quite as good. It was only a little over 80% control. So this, we went out and we tried some trials actually this year with the baculovirus. And I will explain a little bit more of some of the issues that you might occur when you do bee vectoring in commercial trials. Now, one of the issues that we found early on was that when you had very active bee activity going exits, coming out of the hive, going through the dispenser, you were getting caking or crusting over of the inoculum. This was due to fecal and fluid deposition by the bees, and also you can get high humidity. So it was caking over the inoculum, and the bees were not able to pick up as much on their hairs. And this would happen after 4 or 5 days. Actually, we used as our carrier corn flour. And now one of the companies, Bee Vector Technology, has developed a new carrier, which is being patented, which has the corn flour, but also includes desiccant stickers and other ingredients, which eliminated this problem. So this really helps. So this was actually a good investment that was made by Bee Vectoring Technology. Now, I talked about the hives, different types of dispensers. This dispenser you see here, I call it the Sutton University of Guelph dispenser here. This is what we used for most of our trials, and this was in front of the hive. You attached it, and the bees, this is a plexiglass model. The bees would come out, they go into this little box, they would drop into the lower half of this dispenser. They would walk with these baffles here, and then they would come out on the top. There's a little hole here and exit. And then when they go straight back in, they just go straight back in without going through the dispenser. This worked very well, and we're able to put about 20 grams of inoculum in the dispenser, and it will last a week. The next dispenser is the Bee Vectoring Technology dispenser. It's built into the hood, the lid of the hive. As you can see here, this is the tray, and the tray's over here, some bees in it. When we've lifted the lid up here, when it's down, this little space here is actually open to the hive. So the bees come in, they walk around all these pegs. That helps them pick up more inoculum on their bodies. They would then exit, and then they go back into the hive through a separate hole. We found when we compared the VVT dispenser with the Flying Doctor that the bees actually picked up 1.5 times more inoculum using this type of dispenser. Now, this is the Flying Doctor dispenser, patented with BioBest in Belgium, and you can see here it has, again, it has a tray here with where the bees can walk and pick up the inoculum here, and you can see here is another insert here showing the inoculum. And then when they come back, they come out, actually there's 2 exit holes, and this little insert shows you a bee going back in, and it goes back in and follows this entry right here. And we found that the activity, the number of bees leaving the hive, going in and out of the hives, was 1.5 to 2.5 times greater with the Flying Doctor compared to the BVT. Now, one of the problems that we encountered with when we did our baculovirus trials last summer was actually getting some of this inoculum not being picked up, actually going through the dispenser. We only did these trials with the flying doctors, and what was happening here is that there was bees, when they come out of the hive, they weren't too interested to go through the dispenser, and they were going back in through the ventilation holes at the side of the hive. And you can see here a bee going in and some coming out. They would build their nests there and they wouldn't go back through. So we were only getting about 20–30% of inoculum being taken out of the trays. When we screen, put some fine mesh here, just actually not fine mesh, window screening here, we were able to get actually 70–90% of the inoculum being picked up by the dispensers. So this really helped out. So that's why we feel that these trials need to be repeated, and it's important to screen the vents. So things to remember when you're doing bee vectoring. One, the number of hives with dispensers per acre. With the Bavaria, it was essentially 2 hives per acre or 5 per hectare, and that worked quite well. And again, when we did the BT, we used the same rate. But again, it may not be the same for all microbial agents, and it's important to evaluate this and test it. And I think this is something with the baculoviruses, you may need to have more hives that have dispensers per acre than 2. But not all hives need dispensers, and that's important to remember. We find that it's a good idea to screen the hives. I think it really improves what's picked up in the dispensers. Always check the hives for bee mortality. Normally we're picking agents, and it's always important to evaluate to make sure these agents don't hurt the bees, but still you should be checking to make sure these hives are functioning like they're supposed to. And the interesting thing over the last 2 years is we had a grower that had tomato spotted wilt virus in his tomatoes, and he wanted to do bee vectoring. So we did trials there with the Bavaria bassiana, and he was able to control his thrips. Western flower thrips are the important vector for tomato spotted wilt virus. So he controlled the thrips enough, and we were finding thrips on the tomato plants infested with the Bavaria that he did not have any of the disease in his crop. So it certainly did work in this case. Now also, you have to remember, bee vectoring is not a magic bullet. It's to be integrated with the other control measures. And of course, I mentioned that it's both environmentally and economically friendly, and that bee vectoring is generally compatible with the other biological control programs. So that's really important. And I also like to acknowledge, as you can see, this really was a group effort from everybody that supported us in providing funding and other supplies and other institutes that we worked with. Thank you.
Well, thank you very much, Les. I think what you've done is introduce very much the concept of what our technology has done over the years and how it works, and some of the things that one has to keep in mind. It has been indeed a team effort. I just want to go on now with some examples from open-grown crops or field crops, and tell you some of the success stories that we have in there, and perhaps also introduce to you some of the difficulties. The first trials we did were really in the 1990s, and we used honeybees to take a fungal antagonist called Clonostachys rosea out to strawberries in flower and deposit the Clonostachys on the strawberry flowers, and it suppressed gray mold on strawberries and raspberries. And gray mold, as you know, is what causes your strawberries and raspberries to go moldy in the refrigerator. So this was the experimental setup that we had. Our controls were actually in the screen tents, so bees with the biological control agent on their bodies could not get at those flowers. And one can see the hives then here and here and here set up in the field, and then the actual plants that got the biological control agent on the flowers in between in the open. So this antagonistic fungus, then Clonistachys, was delivered by honeybees to strawberries and then onto raspberries and resulted in grey mold control, which was equivalent to commercial applications of fungicides. And that is what we demonstrated back in the 1990s. The experimental setup then gives you an idea on the honeybee hive what those dispensers look like. They are, again, sort of one-way dispensers because the bees, when they go, they come through the inoculum. This tray is pushed in, and then they come up this screen and out underneath this lid and then fly to the crop. And then when they come back in, they land on the bottom board and go underneath this tray to get back inside the hive without picking up any of the inoculum and distributing that into the hive where we don't want it and possibly doing damage. So here's a honeybee coming out of the hive, and you can see how well dusted it is with inoculum. So we continued this work. Dr. John Sutton and Todd Mason and myself continued this work in 2012, 2013. And we got, as you can see here, 90% reduction on commercial fields then of grey mold. That's good. We got a surprisingly 100% reduction in phomopsis. This was not something that we were really expecting, but nonetheless, and a 93% reduction in rhizopus leak. And that also was not terribly expected. So we're not only just going after grey mold now, we're looking at this whole assembly of diseases that get onto the ripening and ripe fruit and cause losses to growers putting it into the market. We used the Clonistachys on blueberries as well to have an opportunity to see if we could control mummyberry and grey mold on blueberries. And here's a low bush blueberry field then in the province of Prince Edward Island, and one of the blueberry farmers there and somebody sort of inspecting the flowers. And bumblebees are very often used now, as well as honeybees, for the pollination of blueberries. They require cross-pollination. So we started experiments in 2009 and 2000 through to 2011, and those experiments are still going on. And this is, as you can see, is like the dispenser that Les showed us on the outside of 4 packs then of bumblebee hives put out onto blueberry fields. Well, one of the things with mummyberry particularly is that mummyberry we can suppress. It moves from the leaves to the flowers on the bodies of pollinators, so we need to break the life cycle. So the mummyberry, actually, you can see the mummyberries here. These fall off the ground in the summertime, sit on the ground, and then in the spring of the year, then the mummyberry fungus produces the fruiting bodies which shoot the spores into the atmosphere. Those spores get onto the leaves, infect the leaves, and those leaves produce, as a result of this, a honeydew. And that's where the bees pick up those particular spores from that honeydew and then transport it to the flowers while they are pollinating. And that is how the life cycle of the mummyberry is complete. So we need to sort of think about this as breaking the life cycle in 2 different places with our technology, and that we have been able to do, but it's complicated, as you might imagine. At the same time, we were able to suppress grey mold on the blueberry fields in the same way as I explained for strawberries and for raspberries. Now we move into a different field crop. This is a sunflower field. We worked with a very interesting system here. These are organically grown sunflowers. And here is a 4-pack of bumblebee hives with the in-lid dispensers that Les showed you. And we were able to put Clonistachys and Bacillus and Bovaria onto the flowering heads of the sunflower with bumblebees. So the bumblebees are the vectoring agents. We were able to suppress banded sunflower moth, because of the Bacillus thuringiensis we used, and also to suppress Sclerotinia head rot. And we ended up totally, for the grower, with a 40% improvement in yield with more and bigger seeds and less disease. So we were getting pollination plus crop protection. We're not able to tease out what of those provided the greatest amount of the 40% improvement in yield, but the total picture is certainly very good. So this is not what we want to see, Sclerotinia head rot, but these perfectly filled pest-free heads with high-quality seeds. Then we did some work with lygus bugs that Les has already introduced us to, and there's a lygus bug on a canola flower using Bacillus thuringiensis, and we did some experiments on that in the field in cages using honeybees, again with in the canola experiment. And here's Mohammed Al-Mazwari, who's now at Al-Balkh University in Jordan, setting up the system. It was part of his PhD program. And here we can see then that the honeybees were coming out 100% with the Bavaria dust on them. About 80%, 75%, 70% of the flowers were getting Bavaria. The leaves were also getting Bavaria, and the tarnished plant bugs in the cages were getting Bavaria as well. And the result of that was that, in fact, we were getting close to 40%, 45% tarnished plant bug mortality with just the single treatment if bees and Bavaria were present in those cages at the same time. Whereas if the bees plus inactivated Bavaria, we heat inactivated it, we didn't get anything like that mortality, and it was the same as if we had done nothing and used those tents as controls. So very, very good sorts of results. This will give you just some ideas, and certainly these PowerPoints will be available on YouTube through the Canadian Pollination Initiative, so don't need to worry about not being able to read everything. But with pome fruits, we've been able to attack fire blights, and more recently in 2014 did some work in Nova Scotia with the growers using bumblebees and using a biological control agent against fire blight. On strawberries, we've attacked grey mold, Phomopsis, Rhizopus with Clonistachys and honeybees and bumblebees. On raspberries, the same sort of thing. Blueberry, again, sunflower and strawberries and blueberries using sort of, and these are the people who have done it in other places. The people in blue are people associated with Les and myself and the University of Guelph. And then also with clover. Originally, Dr. Gross at the USDA did some work with nuclear polyhedrosis virus against Helicoverpa, and was successful in that. And then Tariq Butt in Britain on pollen beetles and metarhizium with honeybees, and so on. So and there's our own work, or work also done with Bavaria using honeybees against the broca, or the coffee berry borer on coffee in Ecuador. Very interesting work, and highly indicative, and we're following up with trying to look at coffee and using biovectoring on coffee, and have experiments now in progress in Brazil and soon to be in progress in Mexico. So the conclusion is that there's huge potential. Both bumblebees and honeybees can successfully disseminate Bavaria to greenhouse peppers. They can reduce the numbers of lygus and thrips in field canola. Also whitefly and green peach aphids in greenhouses. We would expect it to have a similar effect in the field as well. And so we have these things can be co-vectored, as Les has pointed out. And so pollinator biocontrol vector technology is a win-win situation. In fact, it's more than just win-win. It's win-win-win situation. Brings along the benefits of pest control with better pollination, so crop protection, re-entry times into the field are that much reduced, And it looks from our initial work that it is and can be a lot cheaper than conventional pesticide applications. So combining pollination with biological crop protection results in improved yields, larger crops, fewer chemicals, less residues, and so on. So I think it has a number of things going for it, but there are still some things that need to be done, as Les has pointed out. There are some questions to be asked, even though it is now ready and is being used commercially by a select group of growers, particularly with those who have interest in reducing pesticide use on their fields. And of course, that includes the organic growers, but this isn't restricted then to the use by organic growers. So I think that's my presentation complete, and we have about 20 minutes left for questions.
Don't click anywhere. Just answer the question.
Okay. Let me do.
Yes, not further into that.
Okay. The question there, can the successful story of bumblebees and Bavaria bassiana happen for metarhizium? Well, we've actually taken a look with bumblebees and metarhizium, and this was metarhizium anisoplii. I think it's now called brunium. And we found that we're able to show bee vectoring here again, and that we did this with whiteflies. We had about 58% infection of adult whiteflies, but we did a concentration on the inoculum of 5 times 10 to the 9. And that we also found was too high for a concentration level and had negative impacts on the bumblebees. So we need to do more work in that area, and I think that there's been work done in University of Ghent that showed that you need to actually get your concentration down to below, I think probably times 10 to the 8th. At that level, the impact on the bumblebees was not significant. Now, does it work in open fields as well? Well, I think that Tariq Butt and the work that he's done there in canola rape in the UK shows that yes, it can be used in open fields too.
So the question is, does the bee-vectored biocontrol system work only on pests and disease that affect blooms? The answer to that is no, because the pests and diseases that we have targeted and been able to suppress are pests and diseases of the fruit, of the leaves, of the stems, and of the whole plant. What has happened is the bees have taken the biological control agent out into the field or into the greenhouse, and it has become so well dispersed on the leaves, under the leaves, on the stems, in other places, that it has an effect on pests that are not associated with the blooms or even associated with the plants at bloom time. So this is remarkable. We don't understand how the biocontrol agent is getting around. There's obviously some very interesting biophysics associated with that, with small particle movement in the atmosphere, but we have not looked into that except to be able to monitor the biological control agent and find it so well dispersed wherever we have used it. And the biological control agent has originated then in the dispensers on either bumblebee hives or honeybee hives.
Actually, Peter and I both aren't plant pathologists, and they probably be able to answer this better. But what we feel that it may work better. PCR may be a better tool than selective media, because when you use the selected media, there's a lot of work to just preparing your sample, and of course you have to wait for 5 or 7 days before you can read it. So I really think that's an important question. If it's faster and less expensive, I think that would be great.
I think it's very, the question is it right that we cannot count on different publications' results as they use different strains, except the one used the commercial material such as Botanigard? I think one has to read the scientific publications and also perhaps publications put out towards growers with some degree of care and see what it is that has actually been used. We certainly know that Bovaria is effective on a wide variety of pests, as is Metarhizium. Metarhizium is a little more difficult to culture. It has much smaller spores. It's very much dustier, and so that results in some problems in culturing, which is why we've gone with Bovaria rather than Metarhizium, just easier. But yes, there are, and then of course there are different there are different efficacies associated with the different commercial strains of the various biological control agents that have been used. So those need to be sort of looked at carefully. The diluents are important as well, because some of the early work indicated that this technology did not work very well, and people who were doing the work used talc as the diluent, and talc is very irritating to the bees. So what the bees do is they immediately fly out of the hive and groom all the biological control agent off them as much as they possibly can, and then of course there's not much left to go onto the crop. We did not do that. We did tests to see if talc was a suitable diluent, and the results were published that says, no, don't use talc. Use something which is less irritating to the epidermis or the exoskeleton of the bees. So different publication results, one needs to have a look at not just the strains and the actual commercial use, Bovaria, Metarhizium, Trichoderma, Clonistachys, different species of Clonistachys, different species of Trichoderma, etc. One also needs to sort of see how the material was formulated, and also the kinds of dispensers that were used. As Les has pointed out, they have their pros and cons.
Well, are there any side effects or other organisms affected by these services in open fields? Well, I think that the work that Peter's been doing out there, possibly, if the bees go beyond the target crop that they're being used at, there's possible side effects, but nothing has been observed. And I think this is something that it's important, and I think it should be looked at in the surrounding crops or the surrounding plant landscape around your crops that you're targeting to see if there is any impacts, because some of these agents do have fairly broad spectrums. Peter, do you have anything to add?
Yeah, I can add to that we have been able to trace the biological control agents within the fields that we have used, and the biological control agent doesn't seem to get any further than about 150 meters, perhaps a little more from the hive. We're trying to get a little publication written up on that to explore that for a couple of reasons, but the biological control agents don't seem to get that far from the hive when they're in the open fields. And the other thing with the biological control agents that we are using anyway is that they're naturally occurring in the environments anyway, not necessarily the exact same biotype, but nonetheless, Clonistakis is everywhere you go. It tends to be found mostly in the soil if you start looking for it, rather than on the surfaces of plants. And similarly, Bovaria is a naturally occurring entomopathogenic fungus and is also an endophyte in many environments. So we're in a way supplementing something which is already natural, but certainly this question about the side effects is important and will have to be addressed for purposes of registration as the research and development continues.
Well, as far as I know, and that's why we chose microbial agents, is that the pests do not develop resistance very quickly to most of these microbial agents. I know that with the BT, Bacillus thuringiensis, that sometimes the pests can develop resistance, but it is reversible once you stop using it. So I think that like Peter says, we're using the agents that are already out in the environment. So I don't think that this is really going to be a real disaster in the future at all. I think that it's always important to look at your whole ecosystem in the broader sense and to be aware of anything that may happen. But the short answer is no. I think they don't develop. They do not develop resistance, the pests, and this should not be then any real disaster for our ecosystem in the future. Yes, biocontrol agents do require a high humidity to work, not necessarily free moisture. But the important thing to remember here is that the microclimate on the plant, at the plant surface, is much higher than most people understand or think. It could be when you're only a few centimeters away, the humidity level drops drastically right down, but actually at the plant surface, you are getting humidity that is probably 80–90% or more, which is required for the fungus to germinate and to penetrate into its host. And we've done trials in greenhouses where we measured the ambient humidity, and it was about 70–75%, and we were getting very good. We were getting over 80% infection of our thrips and our whiteflies. So on the plant surface, the microclimate is what makes these microorganisms work.
Yes, our results have showed no short-term mortality on the bees, and at the moment we don't know about long-term effects on the bees. We would expect that there would be none, but again, it's one of these things that will need to be tested, particularly with present concerns in Canada and elsewhere over overwintering mortalities, which have been very high, particularly in Quebec and Ontario in places. So we do have to be aware of this, and this would be something that again would have to be demonstrated for use of this technology using honeybees and overwintering mortality. Another aspect of that would of course be human food and residues in human food, which again we don't anticipate that there will be any difficulty with that for the registration process, but nonetheless it's going to have to be investigated.
Peter, you might want to mention that the bumblebees are commercially produced, so that's not an issue.
Yeah, for the bumblebees, it's they are commercially produced strictly for pollination. They have only that one cycle, life-term cycle during which they're used for pollination, and they live and die as pollinators. So there isn't a long-term effect that we need to worry about on the bumblebee front. I think as far as perennial field crops, like raspberries or cherries or blueberries becoming systemically protected by Bovaria, we haven't seen any evidence for this, even though Bovaria can be an endophyte. It doesn't seem to be expressing itself in such a way that it then is having much effect on the mortality of any of the pest species. But we haven't looked at that, so it's a very interesting ecological question. But I would suspect that the mortality caused by Bovaria becoming systemically involved in perennial crops would not be would be very small or non-existent. We have no success stories for use of our technology in forestry pests and diseases. The closest we come to that is our use on apple orchards, where we are seeing some effects. And this was work that was done by Sherm Thompson years ago, working in Utah, and then by Johnson in California for fire blight control or fire blight suppression in orchard trees. And we've been able to see some of the same sorts of things happening in Nova Scotia, where we did experiments last summer. But that is looking good. But at the moment, we're very early on in that research. We did have a look at the other side of the coin of this, forestry pests, particularly thinking about blue stain fungus on some of our pines in Western Canada, and particularly pine forest die-out. Not using the pollinators as the disseminators, but using the using the bark beetles as the disseminators. We investigated that a little bit. We could do that. The problem is, how do you get enough bark beetles all dusted with biological control agents so that when they go out, they suppress the blue stain fungus, which is actually what causes the death of the forest trees. And so there is some possibility there, but whether it would ever be economically feasible or even practical, we really don't know. This would require a lot more work. The question is, have we looked at the impacts of any of the entomopathogens on small hive beetles or wax moths? The answer to that is we have not, but other people have. Now, there's work being done at the USDA lab in Weslaco, Texas, on entomopathogens that have been put into honeybee hives, particularly for the control of varroa mites, and that's showing some promise. I don't know about entomopathogens on small hive beetles or wax moths. We have been interested in whether this would work on wax moths, but we haven't actually conducted experiments on that. And it might be a possibility of interest to the beekeeping community, but at the moment, no. But certainly there has been some work done at the University of Guelph by Ernesto Guzman using Clonostacis, which is not an entomopathogen per se in the strict sense, in honeybee hives for the possibility of suppressing varroa mites. And there's some indication that in fact it has an effect, but it's not a great enough effect, I think, that would suggest that would be an approach to the control of varroa mites. But other pathogenic fungi that would attack the varroa mites could be delivered into the hive this way, as has been shown by the team at Weslaco. I'm sorry, I can't remember the fellow's name down there. I can picture him in my mind, but I can't think of his name. So.
question is, can Bavaria bassiana bumblebee system be used for strawberries? And yes, it can be used for strawberries in greenhouse and open field. And in fact, I know that in open field studies that they did here, they used Bavaria bassiana for aphid control, and they were able to provide some good aphid control. Obviously, it would work against the lygus bug very well here too. So I think that yes, this whole system has been looked at in Ontario, is working very well. Also, you can use the tunnels, and when you're having the strawberries growing in the tunnel system here, it works excellent too. You have anything to add, Peter? Because I know that you've been involved.
Yes, we have done some initial trials with commercial tunnel-grown soft and tender fruit, working with growers in Europe, and indeed this seems to work quite well, not just for strawberries but also blackberries, raspberries, blueberries, and other soft and tender fruit, which are going straight to the fresh fruit market in Europe. Now, obviously, the fruit that we're treating is not going into the market because of registration issues and other things that would have to be overcome. But the initial work is, yes, we get the protection and the extra shelf life, certainly, that would be useful. And so, yes, it's looking very positive, but there are a few more little hurdles to be overcome before that can be commercialized. So, well, thank you very much for all those very interesting questions. Certainly, we'd be more than happy to entertain any questions that you want to send us by email. And those of you who want to spread the word further by spreading the YouTube channel version of this around, please do so. We're only too happy to have as many people as possible aware of what we've been doing. So thank you very much again, and thank you to all our sponsors for the research work that we've done, and also for helping us put the webinar on for you and being able to share this with you.